Okay, so I'm going to be talking today about InterMine as a data sharing platform. So my talk is in two parts. So first I'm going to tell you about InterMine and how it enables data sharing. And then I'm going to give you a specific example about how InterMine can actually be used for using its data sharing properties to do real science. Okay, so who are we? So we're InterMine. We started in 2003. We have been open data advocates for a very long time. We are based here at the Department of Genetics in Goss Miklem's lab. So what exactly is InterMine? It's a data warehouse. And so it's open source. We're on GitHub. Everything is open. And it's just for biological data. So what do I mean by data warehouse? Basically what you can do is you import your data file into InterMine, and then InterMine generates a user interface for you. So you get a website with it that you can put on the internet so your data is instantly searchable. So there is a core data model in InterMine. So when you import your data file, you match your data to the data model. And by data model, I mean the relationships between the data. So like one chromosome has many genes, so that one-to-many relationship. So that's what I mean, the core model. So that's important for data sharing because you're able to search across the InterMines because you know what the data looks like. If your data doesn't match the core model, you can extend it, but every InterMine has a core data model. We also have a sophisticated data integration system, so you can import your data file, but you can also pull in publication data, other data sets into your database. So once you have a database, you can get a user interface for free. You don't have to be a web developer; it just comes with the software. The web app comes with some data analysis tools. So we have some network visualization, some enrichment statistics, and an advanced search. So we have like a Google keyword search so that your data is really accessible to the public. We also have InterMine comes with an API. So what I mean by that is the data is programmatically available. So this means that users who like to run scripts can access your data. So you have your user interface where people can do like a Google search, but you also have the API. So your bioinformaticians can write a Perl script and access the data as well. So it's nice for pipelines. So this is just a visualization. So we have the disparate data into the database, and then out you get the web app, and you get your API and clients. So who uses InterMine? Sorry, that's not readable at all. So we have dozens of InterMine instances that people run. Probably the biggest ones are the biomedically relevant model organism databases. So these are the mouse and the rat. These are, by model organism databases, these are organisms that have been studied because they've been useful for discoveries about human health. So mouse and rat, of course. Zebrafish has been useful for heart things. Fruit fly for Alzheimer's. And yeast, and I'll talk about that in a minute. So what sort of data can you put in InterMine? So it's biological data, any biological data. So genes, proteins, publications, disease. Okay, so how exactly does InterMine enable data reuse and sharing? So the whole point of InterMine, the reason why InterMine was started by the founders was that to make data available and useful to the public. So they weren't happy with how data availability, and so we developed this software solely to make data available and useful to the public. So fair data principles. So the working group from Force 11, which I encourage you to Google, they came up with these data principles. So FAIR stands for findable, accessible, interoperable, and reusable. So this has since been adopted by Elixir and I think the NIH Better Data to Knowledge project. And what it is guidelines for data sharing and data stewardship. And we found it kind of useful. We're here to help data sharing, so we found a nice framework to see how well we're doing enabling data sharing. So findable, so I'll go through these and tell you how InterMine enables these. Findable is the first one, so identifiers. So if a gene, every research object in InterMine has an identifier. So for instance, if you're talking about a gene and a gene is in InterMine, you can use the identifier to make sure you're talking about the same thing, which is really important. InterMine also has a lookup system, so even if you're using what we call a synonym, something maybe that is obsolete, you can still find what you're looking for. Searchable, so InterMine has a lot of search tools. You know, the keyword search, really tools that anybody can use. And you know, InterMine is indexed by Google, so all the data is meant to be eminently searchable. Accessible, so we have a public API, so anybody can access the data. So you have the user interface, you have a public API, it's all accessible. And we use popular languages for our clients, so Perl and Python. So these are industry standards, languages that a lot of people know. Data interchange formats, we use the industry standards. So these are the community standards that everybody works with. So you can download all the data from InterMine in JSON, XML, tab, delimited, comma separated format. So we don't use any proprietary formats, any proprietary languages, no proprietary software actually. Everything is open and standard. Interoperability, so we're really big on interoperability. This is software talking to each other. So InterMines, of course, can talk to each other quite easily because they share a core data model. But actually, it's not too difficult for other software to talk to InterMine, and they do, because we have a public API, so anybody can access all of the data all of the time. So we also use ontologies. Our core model is based on the sequence ontology, which is a community-created ontology. So an ontology is basically sort of a controlled vocabulary. So it's important for data sharing to have a shared vocabulary so that when I'm talking about a gene, somebody else is talking about a gene, we're talking, you know, sharing a vocabulary. And that's actually controversial. And then reusable, so all the provenance data is available. So that's how InterMine enables data sharing. So now I'm going to give you a specific example about how you can use InterMine's data sharing properties to actually do something. So I'm going to use YeastMine to predict potential chemotherapy targets. Okay, so I'm going to show you in this example, I'm going to use a set of genes in InterMine, and I'm going to do some analysis to identify potential chemotherapy targets. So in biology, genetic interactions can show us a lot about functional information, functional relationships between genes. There are a lot of types of genetic interactions, but what we're going to talk about today is synthetic lethality. And I'm just going to quickly tell you what this means. So synthetic lethality is when you have two genes have a mutation, the cell dies. If you only have one mutation, the cell lives. So in this example, so we have a normal cell which has the BRCA gene, so this is the famous Angelina Jolie gene, and PARP. And then we have a tumor cell which has BRCA with a mutation and PARP. So what we do is we add a PARP inhibitor. This causes PARP to mutate. And so the synthetic lethality, because they have a synthetic lethality interaction, the healthy cell, the normal cell, is fine because you only have one mutation. The tumor cell, because you have two mutations, the cell dies. So the healthy cell lives, the tumor cell dies. So you can see that synthetic lethality is really an important way to find drug targets for cancer treatment. Okay, so that's synthetic lethality. So the cell dies. So I'm going to do that. I'm going to look for synthetic lethality interactions in YeastMine. So what YeastMine is, it's an InterMine, using the InterMine software. It loaded a lot of yeast data into InterMine. It's run by SGD in Stanford, so these are the yeast experts. And by yeast, I'm talking about the beer yeast and the bread. So this is the yeast. You wouldn't think that you could help with cancer, but we've learned a lot because it's so simple. We've learned a lot about the cell life cycle, like signaling. The cell signaling genes in yeast are the same, we think, with the cell signaling genes in you. So it is a useful organism to study. So Stanford and Mike Cherry's lab, so they created an InterMine and they put a bunch of yeast data in. So they put a bunch of genetic interaction data in from BioGrid. And so we're going to use the tools in InterMine to search for targets. So the first thing we're going to do is we're going to download from this other database some tumor suppressor genes, 173, from Vanderbilt's TSG database. So data sharing again. So we download 173 gene identifiers and we put them into InterMine. So we have our tumor suppressor genes. These are human genes, but we're going to convert them to yeast genes. So we're via homology. So homology uses some algorithms and identifies functionally equivalent genes. So like I said, cell signaling in human is the same in yeast. So we use homology. We've identified the functionally equivalent yeast genes. So we went from human to yeast. And what we did, and we did this, we're doing all this in YeastMine, in InterMine. And so we have the equivalent yeast genes. And now we use InterMine's powerful search, and we search for all genes that interact with our equivalent yeast genes with synthetic lethal interactions. So we converted the tumor suppressor genes, so we have the equivalent tumor suppressor genes, and now we're getting the synthetic lethal interactors. So these are the genes that interact with our tumor suppressor, and now we get the equivalent human genes via homology the same way. And then we think now that we have synthetic lethal interactions. So this would be a potential, we think, target for chemotherapy research. So we look in our results of our human genes, and we have the human ATR gene and the POLD1 gene in that we think here would have a synthetic lethal with our tumor suppressor genes. So we look, and we actually look in InterMine, and there's a link out to a publication, and there is experimental evidence that does say that ATR and POLD1 do have a synthetic lethal interaction. So there's evidence to support what we just found in InterMine. So this supports the InterMine. The workflow I just gave would be helpful to find drug targets for the treatment of cancer. Okay, finally, so in the future, we are kind of planning to put InterMine in the cloud. So right now you have to set up your own server, but we had an idea where you'd be able to upload your data and it would deploy a web app or user interface to like the Amazon cloud instantly. So click, and then you'd get an interface. And the last thing we're working on, federating search tools. So we have a prototype that's searching a specific data set across all of the InterMines. So that's another future direction. Okay, thanks.
Okay, that's great, thank you. We actually have a few minutes for questions. And gentleman in the second row, if you'd like to. Should
I scream or?
The mic is coming to you.
Yeah, thank you for the nice talk.
Thanks.
I had two questions. The first question, which is about sharing biological data with public and for the industry, private industry.
Yeah.
So would the access of the private industry, to the data subsidize any medications that are then, like for example, cancer drugs that are identified by the open data source for the public? Would there any be subsidizations of these medicines for the public later on?
Right, yeah, I don't know, that's a good question. No, I have no idea. If the data is licensed to be open, so there are different levels of licensing for data. So you can license it. A common one is free for academic use and then charge for commercial. But if the license is to be free for everybody, then no. But we all benefit, right?
The second question, the public, do you see the public engaged into surfing the data?
Yeah, so I guess I had a different definition of public. I guess it was our public, InterMine's public, are biologists and bioinformaticians, and not common, a person on the street, So, yeah.
So, any other questions at all? No? Actually, I have a question. Do people use the data in InterMine to actually publish results? Do they then quote InterMine as the source?
Citation is a big deal. There's some efforts going towards getting software more recognition because publications are not the best way. We do have citations, but not everybody who uses us cites us. So we do get citations, but it's not a relevant metric to show how much impact we have. So usage is. We try to do it, but really, it's hard.
Yeah, no, I know that's a major problem, so
Yeah.
that's specifically why I asked.
Yeah.
Any other questions at all?